Hi everybody, my name is Makhali Voss and today I'm really excited to be here and talking to you about my research titled Flies as Sentinels for Wildlife Disease Ecology, Tracking Histomonas meleagridis and the Black Blowfly Phormia regina. So Histomonas meleagridis is the protozoal parasite causative agent of blackhead disease or histomoniasis. A cyanotic head is not a pathognomonic symptom of this disease and therefore I will refer to it as histomoniasis. With this disease we can see extremely high turkey mortality. In an infected domestic flock, we can see 80 to 100% mortality rates, and we assume these numbers are similar to what we would see with wild turkey flocks, although this remains undocumented. One of the reasons that this is still undocumented is because wild turkey carcasses can be difficult to find. Whether that's due to scavenging or quick decomposition, whatever the reason, this leads to outbreak confirmation being impaired, and sometimes we don't even know that this disease is in an area. Which brings me to blowflies. Blowflies are within the family Calliphoridae and they can quickly locate and utilize carrion. Whether they're feeding and ovipositing, they're also inadvertently sampling any DNA that is present, and it is for this reason that they could be used as potential sentinels for histomoniasis, especially as we know that the flies can acquire the Histomonas DNA from previous research in our lab. My overarching research question is how long does Histomonas meleagridis DNA persist in blowflies? To answer this question, we used a lab colony of Phormia regina, the black blowfly. This species is widespread and abundant in the southeastern United States. We used individualized 3-day-old flies and exposed them to differing treatment types. We had Histomonas-positive liver, Histomonas-negative liver, and water. Flies were exposed to their treatment type for 4 hours under consistent conditions. After their 4-hour exposure concluded, 3 female and 3 male blowflies were randomly chosen and freeze-killed for time point 0. Every 24 hours subsequent to this, 3 female and 3 male blowflies were randomly chosen and freeze-killed until 120 hours post-exposure. Once all flies had been freeze-killed, I performed gut dissections, DNA extractions, and PCRs using Histomonas-specific primers. I then ran a gel, and anything that came back positive and in the size that we were expecting was cleaned up with Exosap and sent out for Sanger sequencing. Once we received the results back, I was able to analyze them, where I then found that we were able to detect Histomonas meleagridis DNA in 36% of the flies that were fed infected liver, and we were also able to detect it 96 hours post-exposure. I then wanted to assess if we saw statistical significance in the Sanger sequences that were Histomonas positive over time, and I found that there was indeed significance. The relevance of this research in a One Health perspective is that these flies may assist with disease diagnostics and mortality events. This is all the more important with this disease because, as I previously mentioned, sometimes we don't know that this disease is in an area, and unfortunately at this time there are no preventatives and no treatments for this disease. This is a perfect example of where rapid response is really crucial within wildlife disease. Also, the persistence of pathogen DNA in flies can help us with pinpointing the timing of outbreaks. If we were to test wild flies and they came back positive, then we would know that within the last 96 hours there has likely been an outbreak of Histomonas in this area. Through using blowflies as a sentinel of wildlife disease with not only this disease, but others as well, we can mitigate the stress put on vertebrate animals during pathogen testing as contact with them is no longer needed. Next steps with this research include using these methods with other wildlife diseases and differing blowfly species. Thank you for your time.